Do it? Okay. Okay. All right, we're going to go ahead and get started. It is my great pleasure to introduce Dr. Chris Schmitt to everybody. Chris did his undergrad at University of Wisconsin-Madison. He did a master's and PhD at NYU as part of the NICEP program. Then he did his first postdoc at UCLA at the Center for Neurobehavioral Genetics. After that, he spent a year as a lecturer at USC and then did a second postdoc at Berkeley with Leslie Lubusko, which I can't say. And he's now an assistant professor at Boston University. He's part of the Anthropology department and the biology department there. He runs the Smeagol Lab, the Sensory Morphology and Genomic Anthropology Lab.
We did contort our names just so we can call it Smeagol.
And it's a joint lab that he runs with Eva Garrett. Chris is going to tell us a little about his work himself, but very generally, he works on understanding the interactions between genotype and phenotype, particularly within the context of growth and obesity. And he's also just a fantastic guy. And I've been lucky to know and work with him for the past several years. So we're very grateful that you came out to talk with us and looking forward to hearing what you have to say.
Thanks, Katie. Yeah, thank you for the great introduction. Like Katie said, I'm really interested in genotype-phenotype interactions. I'm mostly talking about my obesity program at Vervet today, but I just wanted to give a shout out. As she mentioned, I work with Leslie Lubusko at Berkeley, and she does a lot of really interesting work finding genetically patterned dental phenotypes, and that's another research program that I have that I will not even allude to in this talk. But if anybody's interested in that work, I'm happy to talk about it later. I suspect it's not really up people's alley here, though, so we'll talk about this today. So I'm mainly going to be talking about some work that I've been trying to do in establishing vervet monkeys as a model for the evolution of obesity and growth. And part of the impetus for this research is, of course, the obesity epidemic, which is not just in the United States, but for funding purposes, I'll talk about that. Where about a third of the US adult population is defined as obese, at least based on BMI metrics. And it's increasing every year. So if you go to the CDC website, you can download yearly counts of obesity stats, and it just keeps getting, darker and darker as we keep going, at least in how they draw the graphs. And this isn't just in the United States, it's throughout North America, although Canada is doing a little bit better than we are. And this is also worldwide. So for example, in Saudi Arabia, over 70% of the adult population is at least medically considered to be obese. And it's not just adults, about 70% of US children are also considered obese, and this is turning into something of a developmental problem, at least within the healthcare system in the US. And it's really expensive. About $147 billion were spent on healthcare related to obesity and cardiovascular disease associated with obesity within the United States. So it is something that there's a lot of interest in kind of taking care of from a biomedical perspective. But interestingly, it's not only humans that are becoming obese. We also see this in livestock populations, in domesticated animal populations, in lab populations. This is also increasing. So there's really kind of a vested interest in finding out why this might be the case. And I wanted to give a caveat before really kind of getting into it, because I know other people in this room work on obesity in some way. And what I've been finding in reading the literature, especially jumping from primatology to the Center for Neurobehavioral Genetics, where we're working with biomedical populations, and now increasingly reading human biology literature, is usually when we're talking about obesity, especially in the news, we're talking about BMI, body mass index. But then when you're getting into the genomics literature, what you're usually using is all of these clinical indicators of obesity that are really easy to collect in a clinical setting. So you'll look at waist circumference, you'll look at BMI, you may just look at body weight. And these are all of the traits that people have been using to kind of get an idea of polygenic contributors to common obesity. And I'll talk about that in a little more detail in just a second. And then in the rest of biology, like looking at evolutionary biology and human biology in particular, we are getting a little more specific with what we're talking about when we talk about obesity, right? So we'll use some of these same measures, but we'll also look at skin fold thickness. Dual emission X-ray absorptiometry is a great way of finding where adiposity is actually occurring within the body and being able to quantify that through images you get through DEXA. Measures of adiposity differentiating between omental or visceral and subcutaneous fat, because these do have different metabolic functions within the body. Insulin processing, metabolic disorder more broadly. So there's a lot of ways that people can look at this, but usually when I'm talking about the genomics of obesity, what I'm really talking about are these really rough metrics. And even though BMI has been shown to correspond to various kind of sequelae associated with obesity and metabolic disorder, like insulin processing, it's still kind of a rough metric, right? So like what I tell my undergrads when I'm teaching about this, I'm like, okay, well, if you know somebody at the gym who's really swole, right? They've got like really bulky upper body, their BMI could be the same as somebody who's considered to be morbidly obese, just because their body weight for their height is actually much higher than you would expect according to the population metrics. So there's a lot of kind of room for misinterpretation within here. And I just want to give that caveat to begin with because, you know, I'm using some of these same measures, so it's important to understand that we're starting out on kind of unstable ground. And it's interesting too, because when we're kind of digging into these phenotypes, there's a lot of literature that we can approach in order to understand it. And one thing that really isn't in dialogue with this literature, from what I've found so far, and you know, correct me if I'm wrong, I know a lot of you are thinking about this too, is we do have a lot of evolutionary frameworks in place about body size kind of writ large. But this literature doesn't often interact with ideas about obesity, so to speak. Even though work that's being done now trying to link these literatures together is actually proving to be really promising. So I'm sure we're all familiar with some of the classic natural selection literature about body size as we understand it across latitudinal gradients, like Bergmann's rule, this idea that animals get, you know, larger or more voluminous, I guess we could say, as you approach higher latitudes or higher altitudes, presumably to select for greater kind of homeothermy. And then Allen's rule, Wrench's rule, is this idea that as animals become larger in body size, their sexual dimorphism also becomes more exaggerated. And this is something that has been noted in a lot of different species. Although within primates, if anybody's familiar with Adam Gordon's work, it's a little more complicated than that at smaller taxonomic levels. We also, of course, have all of the sexual selection literature, which I'm not really going to get into today, that can intervene with body size variables. And then, of course, there's all the literature on plasticity, which I think people are most familiar with here, like Katie's work in microbiome. There's been really interesting work looking at how microbiome actually kind of co-corresponds with, say, Bergmann's rule in mice, finding that, you know, these latitudinal and altitudinal gradients lead to higher body volume is actually also corresponding with Bacteroidetes and Firmicutes distributions in the way that you see in obesity. Okay, so it's possible that these kind of plastic phenotypes that we associate with variation in obesity traits are actually also implicated in these larger kind of body size systems that we've been thinking about, but not in this way. And of course, there are more obesity-specific evolutionary frameworks like thrifty genotype and thrifty phenotype hypothesis. We're all familiar with this, yes? Okay, so I'm not going to belabor this point in the talk today. But even within these kind of frameworks, there's really not very good integration with this kind of classic evolutionary ideas, and then the more biomedical frameworks that we run into in the human literature. And so within these biomedical frameworks, a lot of really interesting work has been done. This is a classic figure from 2012, looking at the obesity genes that we knew about during this time, at least for complex polygenic obesity. And what we're seeing here is basically all of the genes that have been found depending on the phenotype that we're defining obesity by. Okay, so this is again getting towards this idea of how are we defining this. In blue here we've got BMI. This pink circle up here is waist to hip ratio adjusted for body weight. We've also got waist circumference, we've got body fat percentage, and we've just got obesity writ large. You can see that there's, you know, one kind of fulcrum over which everything is kind of tilting. We've got FTO, which is the ortholog of the OB gene in mice, if you're familiar with that system, that first obesity gene found. We've got a region near MC4R, the melanocortin 4 receptor gene that also is implicated in some forms of monogenic obesity, like hyperphagy, but we see it popping up in complex obesity as well. And then a whole bunch of other loci. And is anybody familiar with this figure? Do you know how much variation in body mass this whole group?
The log GWAS, it's 2.7%.
Yeah.
And that's 97 hits.
Yep.
Something like that.
Exactly.
Yeah.
So like the more,
I'm sure it's less here.
it's less here, yeah, exactly. But the more recent numbers,
Yeah.
it's not a lot.
No.
And the FTO gene accounts for like what, 0.034 or something?
0.45, 0.45.
Yeah, exactly. So I mean, we have obesity genes, obesity alleles, but they're really not contributing all that much. There's a lot of missing heritability in here. Because if you actually do quantitative genetic assessments, it's somewhere around between 30 to 40 to 80% in some cases is supposedly genetically controlled, but you know, we're getting almost nothing out of the genomic signal. So there's a lot missing in this biomedical framework, and in part it's because we need such large sample sizes to do GWAS. We can't possibly get all of the intervening covariates or control for them across all of these populations that we need to get statistical
And if I
power.
could just break in. So
Oh,
part of
please,
what's
yeah.
going on, I understand the point of this is that they're not all overlapping,
Yeah,
but the fact that some of these have such small numbers of genes is likely just power, right?
yeah,
Right.
exactly.
So like you have better measures that have fewer hits because they're smaller samples.
Yeah,
Okay.
exactly.
All right.
Yeah, so this is part of the problem too. I mean, there are presumably ways that you can kind of dig into these regions with more power if you're constraining what you're looking for.
Yeah.
And these are all kind of like wide shot whole genome analyses, so there is very low power. Like the
Yeah.
threshold is 10 to the minus 8 something, right?
Right.
For genome-wide significance. So presumably if we had more controlled populations and we're looking at more targeted regions of the genome, we could get a little more power.
Yeah.
But not many people are doing that right now. And so part of this missing heritability, I think, is also kind of in part because we're not controlling for these environmental or developmental variables. So in the human literature too, and I don't want to like bash the human literature because everybody is working on humans, right? But in the human, let's say the human biomedical literature, maybe to be more specific, the funding cycle is what, every 5 years, right? And so there are a very limited number of populations. You know, there's a couple in this room that people are working with, so that's great, where we can actually follow people longitudinally. And within the context of the biomedical literature where we have limited longitudinal studies, we are finding some interesting kind of critical periods during which childhood diet is having an implication for later onset of obesity. We've got these kind of like really popular examples where we've got, you know, maybe a little bit of hand waving going on about the developmental origins of health and disease framework within the Dutch hunger winter and other kind of really popular population examples. And then we also know from biomedical literature that in utero maternal programming is something that is presumably happening, although again, we don't really have good mechanistic ideas about how this is working. We say epigenetics, we say there may be some kind of markers happening here, but we don't really have good evidence of that yet, even though it is pointing in that direction. And this might be one of these areas wherein that signal is being attenuated when we're actually looking for molecular markers that might be implicated in this. And so, you know, this is all a bunch of, like a huge morass of literature that isn't really interacting very well, at least from how I've been reading through it. So one of the things that I'm trying to do is how to tie all of this together using a primate framework, because I think that we've got a good framework in place with the vervet monkeys that, of course, isn't meant to replace human studies, because, of course, that's a good way to actually understand human obesity is looking within human populations. But in the case of these non-human primates, they reproduce at very high levels. They can be controlled. They can control for their environment in captive setting. But then we also can see a lot of variability within the wild setting. And I kind of first started thinking about this when I started my postdoc at UCLA. And this postdoc was kind of the inaugural coming together of this International Vervet Research Consortium that I continue to be part of. And what this consortium does is takes advantage of this really interesting population history that we have for vervet monkeys, where we have a lot of research being done in the wild setting. We've got a bunch of subspecies that have very different ecological settings In some of these settings, they're really interacting with human environments quite a bit, because vervet monkeys are kind of one of those garbage monkey species, like capuchins in a way, where they can survive in almost any environment and they can eat human food, and they actually do really well in cities. So we can get really good kind of sedentary anthropogenic environments with the vervets, but we also have a lot living in more ancestral kinds of environments, like game reserves or nature reserves across their range. And on top of that, we've got really great latitudinal ecological variability. In South Africa, it goes below freezing. If anybody's familiar with Rich McFarlane's work up at Madison, he's really interested in kind of thermoregulation in these very cold weather populations. But then we also have West African vervets that are essentially living in 110-degree weather year-round. Even more interesting than this, though, where we've got a large amount of genetic variability, a large amount of phenotypic variability, is that during the transatlantic slave trade, a bunch of vervet monkeys were brought over to the Caribbean and they kind of took root there. So if anybody's seen those videos of the vervet monkeys stealing people's alcoholic beverages, this was like really popular a couple years ago. This is happening in St. Kitts and Nevis, and these are heavily sampled populations. We've trapped about 1,500 monkeys just from the islands there. They're also on Barbados, where we've done some sampling. And so because of this severe bottleneck that happened about 400 years ago, followed by this population expansion on the islands, we've got a really good subset of the variability with really broad regions of linkage in the genome, which allows for relatively easy discovery of genotypic variants that correspond to phenotypes that we're seeing in that population. On top of that, in the 1970s, Lynn Fairbanks and colleagues brought about 75 of those vervet monkeys over to UCLA and started a captive colony. That colony in 2008 has since been moved over to Wake Forest University in North Carolina. And this is about a 9 generations deep captive colony that's very well pedigreed. And that gives us even broader linkage regions, even better resolution for finding genomic regions and genomic variants that might correspond with various phenotypes of interest. So today I'll talk a little bit about some of the work we've done trying to understand the variability in body size that we see in the kind of wild environments that we see the vervet monkeys in. And I'll talk a little bit more about how I'm working with the vervet colony in order to understand genomic variability and how that feeds into obesogenic phenotypes. And my goal eventually is to kind of pull these two systems together to get a better understanding of how genomic variability feeds into obesity and how we see that genomic variability responding to these different ecological challenges in the wild, so that we can actually get a better idea of actually testing some of these hypotheses for the evolution of obesogenic traits. And by the way, I didn't say this earlier, but please take a note from Chris. Like, feel free to interrupt me if you have a question or anything. Like, we can keep it casual. Right? So this wild vervet sample has so far been primarily concerned with variation in body mass and growth. So these are samples. Each black dot here represents a region that we've sampled. This is from a paper that came out just this year in AJPA, looking at animals that we trapped across their range. When we trap them, we collect many, many different phenotypes. We've trapped 2,281 individuals so far. This is largely cross-sectional data. However, in certain field sites I've been returning to over and over again, we are actually getting some longitudinal data. We've trapped in all these different countries, but the countries in bold are countries where we've done some very deep sampling. So we've got a sample of at least 150 animals in each of these regions. In the case of St. Kitts and Nevis in South Africa, we've got almost 1,000 from each of those areas. And we're really doing very broad phenotyping within these samples. So we, depending on the region and depending on local permitting issues, we've collected all of these different phenotypes. I'll mainly in this talk maybe be talking about DNA and RNA, a little bit about fecal hormones, morphometrics and body condition, definitely talking about alluding to microbiome. This is work that's done in collaboration with Katie when the permits work on. And in this case, all of the animals have been tagged with avid microchips. These are the same microchips that you use on your pets in order to track them. We put it kind of subdermally on the neck, and this way when we trap them, we can scan them and then build up a longitudinal database, at least at the sites where we do retrapping. And this photo here is just us with our veterinarian and a couple of BU undergrads. This is kind of the assembly line. We anesthetize the vervet monkeys in drop traps. The vet takes care of them. They're down for about a half an hour, and we collect everything that we can. So we also, of course, have plasma and serum. We have various types of preserved fecal samples. if anybody's ever looking for a data set, this is all publicly accessible, so just get in touch. And as I mentioned, with these samples, we're also collecting morphometric values. These are morphometrics that I've collected myself, so there's not much inter-observer variability in this case. And we try to construct growth models insofar as we can. These are just growth models, basic like low-S regressions across dental age categories. And I understand that dental age categories are kind of wobbly. I know that they're a little iffy insofar as what we can actually get from them, because there's going to be a lot of variability about when different animals reach these dental age categories across actual chronological age categories. So we don't try to extrapolate too much from these. But when we see them, what we do notice is that there are actually very different developmental trajectories between East African taxa, which are reaching a relatively small adult body size, and West African and South African taxa, which are getting much larger in adulthood. And in this case over here, we've got females, here we've got males, so there is sexual dimorphism in this taxon. And so this variability that we see was interesting when we were kind of doing our summary stats, so we decided to dig into it a bit. And again, this is from a paper published with Trudy Turner, who lives like 10 minutes away from here, and I forgot to get in touch with her to let her know I was here. Published in AJPA, but I think we have a question, Stacey.
Yeah, quick clarification question. Are those animals also longer too, or are they is their body mass just
That's a
bigger?
great question. They are not longer. Like, if you actually look at crown to rump length, they're all exactly the same length. So a lot of this variability in body mass is variability. I can't say whether it's between muscular variability or body fat variability. We just don't have that capability. We do have skin fold thicknesses for some of these, so we could suss that out a little bit. But yeah, I'm not sure.
They're not longer.
Yeah, they're not longer. Yeah.
And what you're talking about here is this sort of clump of the green and a clump of the sort of blue.
Yeah,
I
exactly.
mean, it's not super clear, but there is a sort of clustering.
There is a sort of clustering, yeah. It'll be a little more
It looks
clear.
like it's clearer in the females than males.
It is, yeah. The males, we have a lot more variability, but I'll talk about that in a second too. Yeah, cool.
And do you have limb lengths? Do you have body proportionality data as well?
Yeah, we do. So I don't have any slides about that today, but we do address that within the paper. So just for you, I'll talk about that. so we'll get into that in just a second. So within these phenotypes, we were testing, again, these kind of really, I don't want to say basic, but kind of foundational ideas about how we would see body mass varying across these large latitudinal gradients. But we also recognize that, from work by like Alberts and Altman's 2005 paper, various other work people have done, that when these animals are eating a lot of human foods, they get bigger, right? They grow faster in some cases too, although the power for that was really small in that paper. But here we got a growth model. This is just the larger population. So this is South Africa, Synchytson nivis, East African Pygeurethrus, which is also called Hilgardii, and then in red we've got Aethiops in Ethiopia. And here it's a lot more visible, these kind of differences that we're getting, not only in eventual adult size, but also in perhaps the way that they're growing, okay? And this kind of spike in growth around what would be pubertal age within these monkeys is really interesting in Synchytson nivis, and we'll get to that again in a second. So within female body weight, at least, we do find significant latitudinal signal, exactly what we'd expect. As you get to higher latitudes, female body size is getting bigger. And this is across all of Chlorocebus within our large 4-populational sample. Okay? Developmentally, we do see significantly heavier body weight in the South African Pygeurethrus, which, we can wave our hands and say it makes sense. It's colder. The babies need to be bigger. They need to preserve body heat. Although, Rich's work would say, oh, they should couple with their moms then. So, we would have to kind of look at that data set as well. And we see consistent growth, whereas in Synchytson nivis, we've got this kind of weird pattern. And again, because we're looking at dental ages, we're not sure if this is a shift in when these teeth are erupting in that population. That's certainly possible. It's like a very kind of like founder population. Maybe things got really weird. But if their teeth are erupting at the same time, this represents a really kind of stark growth spurt happening at puberty, which is a different kind of developmental trajectory than what we see in the others. But again, I don't want to put too much weight on that. These are just low-S models. We're not really digging into that very well within this sample. But another thing I want to point out is the human impacts are having a really big effect on body size too. So it's all well and good for us to talk about this latitudinal gradient, but we also have to talk about the much bigger effect that we're seeing based on whether or not they're provisioned by human foods or not. And what we're seeing here is we came up with this human impacts index. This was actually made by James Pampish, who now studies human chins, apparently. But when he was a master's student with Trudy Turner at Milwaukee, he came up with this index kind of aggregating all of the garbage dumps and crops that were available, how close they were to highways. And we used that impact index on all of these sites, and did find that when you bin it into low, medium, and high, and in this model we use the medium impacts as kind of the baseline, with low impacts you're getting much lower body mass, and with high impacts much higher. And both of these are significant. So proximity to humans, availability of human food is also an intervening variable. Now, when we looked at male body weight, it was a mess. Okay, like we saw this and we were like, oh, okay, maybe it looks a lot cleaner. Like we don't have this weird Sankitson-Nevis deviation. Maybe we'll find something, but no. And like the R-squared value for this model was like in the 0.0 something range. And so I was like, oh no, now I can't publish this when the females look so nice. But I had a thought. I was thinking, okay, well, we've got all these different models for understanding body weight. One of them is sexual selection. So I'm not really accounting for that in this model. So what if I just take out the most sexually dimorphic species? And when we actually look at sexual dimorphism across these different Chlorocebus populations, we do find that within Sankitson-Nevis, they have the most exaggerated sexual dimorphism. Why that is, we can't say. Most people historically have said, oh, vervet monkeys, they do just what they told us in East Africa when they were studying them, even though this Aethiops population appears to be largely derived now that we look at the genomic data. So it's unclear to us whether or not there's something behaviorally different happening to make this sexual dimorphism happen. But when we take the sebaceous males, these Sankitson-Nevis males, out of the model looks great. We find latitudinal variation exactly what we would expect. We find a very, very small signal from altitude. I don't know how much we can trust this. It's tiny. We're not finding an effect of human impacts, though, which is interesting, to me at least. So that removal of sebaceous is great, and it's just kind of a cautionary tale for when you're doing large-scale taxonomic comparisons, that you need to actually take into account population-specific potential factors that are leading to variability within this trait. Because if I hadn't taken into account this sexual dimorphism exaggeration in sebaceous, my model looks like crap. But if we take it out, we actually can see what we're looking for. So this sexual dimorphism is actually interrupting the signal that we would have found otherwise, if that makes sense. Yeah,
What is
Stacey?
the range of the dimorphism that you're seeing across all populations?
So in South Africa, it's the smallest. I don't have it on a slide in this presentation, but it's in the paper. So in South Africa, it's about 1.1. So the males are slightly bigger than the females. In East Africa, it's a little bigger. It's like 1.3, but in sebaceous, it's 1.7 or 1.8.
Oh.
Yeah, so it's actually quite exaggerated.
So, do you have a hypothesis as to why the human impacts are not playing into that? Because,
No.
like you said, it is surprising. I mean, there's the Amphiceli findings, right? With
Yeah.
the trash dump, where the provisioned animals get much bigger, and also the dimorphism goes up. And you said the human population.
Yeah. And in the baboon example too, the males had much more exaggerated effects from the dump provisioning. And that's what I expected to see here, was like a more exaggerated impact from human impacts, but like we're not seeing it. And so, I kind of struggled with how to interpret this. In part, I wondered if this sexual dimorphism signal is also intervening here. I also wonder if males are maybe migrating across different human impact gradients. And so maybe they're growing up. Like we see this in the area where I'm doing intensive work, like in the game reserve where they're not provisioned, males are migrating in from farm areas. So they're growing up in a provisioned environment and then winding up in an environment with no provisioning.
Mm-hmm.
So I almost wonder if the pattern of philopatry in a species is interrupting the signal.
So you would pick it up if you got them right at the beginning of adulthood, and then it kind of disappears because they move to a different region. Yeah, I suspect that might be why this is happening. But that's like, I'll wave my hands about it, but I don't have any evidence. It's surprising. It's interesting.
Yeah. So at any rate, so this is kind of the big takeaways that we have from this data set. Stacey, yeah.
One more question about exactly that. This seems like an extreme long shot, but what do you know about the differences in the diets of the human populations that live in St. Kitts and Nevis versus?
Yeah, that's a really good question, actually. I don't know. I know that
is there any chance they have way less access, even though they have high human impact scores, they have way less access to the kinds of foods that we typically associate with?
Yes. So Carrie Dorr, who was also Trudy Turner's student, she does a lot of ethnoprimatology in St. Kitts.
Mm-hmm.
She might be able to address it in the St. Kitts context. And we have a book coming out in January, like looking at vervets kind of holistically across all the different research groups that have been investigating them, and there might be a little bit of an answer towards that in her chapter within that book. But we don't have that information necessarily for these other contexts. Like Trudy, in her work that was published in 1997, looking at differences in body size within vervets just within Kenya, she did find that there was a specific provisioning effect in that population. We know that there's a provisioning effect within South Africa, which I'll talk about a little bit later today. But like, I don't know how comparable that is. Like, we're not doing any nutritional ecology that can really address that. That's something I hope to get a student to do sooner rather than later. But for now, I can't really say. We know roughly that in St. Kitts and Nevis, they're eating like sugarcane, they're eating various other kind of like root crops that people have, and Carrie's data shows that. We have rough differences in South Africa. In KwaZulu-Natal, they're eating sugarcane, whereas in the Free State, they're eating like maize and maybe some other crops. But yeah, like I wouldn't be able to quantify it, unfortunately.
Right.
Yeah. Okay, so that's what we have for Bergman's rule. And to appease the gentleman in the back about Allen's rule. So for Allen's rule within this paper, we did do some beta regression because we just looked at the proportionality of various segments of leg lengths. So we looked at like the upper leg, the lower leg, and the foot. And what we ended up finding was that there was some significance, but the beta value was really small. So we found like technically statistical support, but like the beta was like 0.0 something. So there's something happening. The kind of larger takeaway from it was that the East African Pediatrus hilgardii population has stubby little legs. And we don't know why that would be, because that's contra to what we would expect. Yeah. So we wave our hands a little bit about possible biomechanical advantage in juvenility. We don't know what kind of substrates they're using that might be different from what they're using in other populations. But we kind of were like, somebody go look. That's all we can really say about it. But yeah, Allen's rule was like not really supported because of this kind of one population, again, having something weird going on that we don't understand. Yeah. So we also, of course, Bergman's rule is just supposedly a proxy for climate variation, right? So we went in and we took some data from the World Climate Database in the raster package in R, tried to correspond these variables, instead of doing the regression with latitude and altitude, doing it with these environmental variables. And we found no significance in the females. And we don't know if this is because the World Climate Dataset isn't capturing local variability that's actually important to these body measures. We just don't know why that's happening. But it seems to run contra to this idea that this latitudinal signal is because of various ecological gradients. But again, we're restricted by what World Climate is giving us, so it's possible that we're just not capturing it. With males, we found a lot of significant results. Like body weight was lower with increased seasonality, higher with precipitation seasonality, lower as minimum temperature of the coldest month increased. This last one makes sense, right? Like it gets colder, they get bigger. But these other ones, like my conception of seasonality is based on North America, so I think, oh, highly seasonal, colder winter. But seasonality may be something different within this data set. So I'll need to dig into it a little bit more to understand what's happening there as well. So overall, this analysis led to more questions than answers. We don't really know what's mediating Bergman's rule in this case. We would need much better, more specific climatic data to really suss that out. We don't know what's happening with Sebaeus. We think sexual selection, like greater dimorphism. Maybe there's more direct male competition for mates. We do know that in Sebaeus, and this is another hand-wavy thing, if you're at all familiar with Jennifer Dancy Kramer's work, she was also Trudy Turner's PhD student. She's found, looking at scrotal coloration within the vervets, I don't know if you're familiar, but they have these bright, vivid blue scrota, and they do something called like a red, white, and blue display, where like their penis is bright red, their scrotum is blue, so they'll like get a boner and like stand like this, and it's supposed to be a signal for females. We do know that previous work suggests that these blue scrota are not necessarily a sexually selected signal. Melissa Gerald actually spray painted vervet scrota different colors to see how females would respond. Females generally were really interested in scrota that were just different. They weren't so much interested in the blue coloration. So generally speaking, we don't think of that blue color as like a sexual skin the way that we do with red, right? Because red is giving us like non-cheatable signals that females can use to suss out male quality. We don't think that's happening with blue scrota, but we don't know, right? A lot of people have criticized Melissa Gerald's work and said it's not very rigorous or good, so it's possible that that's the case, that the signal wasn't picked up. But what we do know from Jen's work is that even though within Africa, in most of Africa, we have these blue scrota, in West Africa, and in the Caribbean especially, rather than becoming more vivid blue as they age after puberty, in West Africa and in St. Kitts and Nevis, they blanch. So rather than being blue, the scrotum becomes white. It's possible, again, hand-wavy, that this is kind of a swap in sexually selected signals. Maybe the loss of that blue led to the need for greater male-male competition because females were no longer sussing out males based on the signal, so males needed to get another way to distinguish themselves to females. But again, like, it's a wild guess. We don't actually have data to address that. And then finally, like I mentioned, this South African form of Pudorethris is the least dimorphic, and that's in contradiction to what we think with French's rule. They have the largest overall body size, but they have minimal dimorphism for this taxon. Why that should be, we don't know. Maybe there's a minimum body size threshold for females to survive in the cold, and so they can't get smaller relative to the males. This is something that we again, don't have really good evidence for. So lots of interesting questions that maybe could be addressed with closer looks at all of these taxa, rather than this kind of scattershot trapping approach that we've actually been using so far. So bringing in the genomics, we have done whole genome sequencing on about 150 of these animals in the wild. We do have a really good vervet reference genome through work that's been done by UCLA and the Gregory Mendel Institute and the Genome Institute in Washington University, which has been renamed to something, I don't know, maybe Katie knows from when she was there. It's got a different name now, apparently. And also McGill University have all worked together to make this reference. The main reason why we really like this reference in comparison to other non-human primate reference genomes is it's got really high contiguity. So I mentioned before that these really broad swaths of linkage that we find in the captive colonies is really beneficial for finding genomic regions of interest. The fact that we have really high continuity really allows us to be confident about these broad linkage regions, so it's really good for gene discovery. And for population genomics, there has been some work done, mainly by Mike Montague at UPenn and Hannes Spardel, who is at the Gregory Mendel Institute. But now this is kind of being taken over by my lab, because Mike is now working on the Cactus Santiago macaques, and Hannes has moved on to Lake Victoria cichlids. Why not? So we've had some work done here, mainly trying to understand broad-scale variation within the genome. And in a paper that Hannes published last year in Nature Genetics, we did find some evidence of genome-wide selection related to viral immunity. The main reason why we were doing this broad sampling in the first place was to look at variability in SIV and development from SIV infection to AIDS. We did find that these different populations adapt to SIV in different ways to prevent them from advancing to full-blown AIDS, which is interesting. And we do see signs of that selection when we look at the whole genome. But we have all of these sequences, and we've looked at almost none of it, and the VCF files are online for free. So, you know, if anybody's interested in looking at genomic variability in vervet monkeys, it's there. And my lab is mostly kind of digging into this now. So what I want to do next is take you from this broad scale variation down to the least variable end. Again, where we can actually have really good power for detecting the phenotypic effects of various variants within that population. So this reflects work that I also did through my postdoc at UCLA, but only published very recently, looking at the vervet monkeys in captivity. These have been shown by Kavanaugh and other scientists to be a very good model for human-style obesity. So we find that when they become viscerally obese, especially in relation to waist circumference, that we see various sequelae that are almost identical to what we see in humans. They develop type 2 diabetes, they develop various cardiometabolic disorders that are comparable to what we find in humans. So we do think this is a good physiological model, phenotypically at least, and endophenotypically, so everything going on inside the body, for what we see in human obesity. And on average, every year, about 10% of the individuals within this colony do become obese, again, based on waist circumference. And so people recognized this early on. They were like, these monkeys are getting fat. And so when the colony was still in UCLA, they were like, we need to make sure that they get a little healthier. So they switched their diet to a high fiber, high protein diet, just with the goal of the whole colony losing weight for health purposes. Now, I remember when I first heard about this, I got really pissed because I was like, you didn't keep a control group? You just changed the diet for everybody? This wasn't, and they were like, no, it wasn't an experiment. We just wanted them to lose weight. Which actually turned out to be really good for the kind of analysis I was doing. So they were fed regular monkey chow up until 2004, at which point they switched them over gradually to this high fiber, high protein diet over a course of a couple months, and they were fed that diet for about 4 years. When they moved them to Wake Forest, they switched them back to chow. I don't know why, maybe it was cheaper, who knows. And overall, this high fiber, high protein diet essentially gave them lower energy, in part because when they first switched it over, they hated the diet, they just stopped eating. But then eventually they got hungry, so they continued to eat. But this high fiber diet actually gives lower available energy. They have to burn a little bit of energy to get energy out of it. So we did see about a 10% reduction in body weight across the colony on average when they were eating this diet. Yeah?
And are you able to determine then what the caloric dip was? I mean, so clearly this wasn't an isocaloric quasi-experiment.
Mm-hmm.
It was an actual shift in the composition as well as a reduction in total caloric intake.
Yeah, and we don't have a good way of measuring that. We tried putting microchips into their arms and then a sensor on their food dish. But they're circuitous. They'll just shove their cheek pouches full of food and run away, and then they'll pull it out and somebody will steal it. So there were a lot of variables that we couldn't account for in order to understand how much of a dip this was on average per individual. So we don't have that information. When this paper was in review, people were kind of like, how did you not do this? And I was like, I don't know, man. This was in 2004. I was still in undergrad, essentially. So we don't have a good answer to that, unfortunately. And we don't know what component of this diet is actually causing the effects that I'll talk about just now. Okay. So again, this set up kind of a natural experiment where we could see different ages at which individuals were first exposed to this diet. And since vervets generally take about 4 years to reach maturity, those individuals who were exposed first during gestation, when their mothers were pregnant, were then essentially getting a whole development of this shifted diet up until adulthood. We have other individuals in what I'm calling the first postnatal periods. These are the first 2 years after birth that were first exposed during this time period. I did try to break it down into those that were just nursing versus those post-nursing, but the power gets very, very small. I'll talk about that in just a second. We have many monkeys, 140, that were first exposed during adolescence. So the 3rd and 4th year postnatally, and then about 205 animals only exposed when they've already reached full adult size during adulthood. So my goals with this were kind of to assess the genetic and environmental components of variation for adult obesity-related traits, to do some quantitative genetics on adult obesity, to characterize growth that leads to obesity, so I did construct much more rigorous growth models than what we can do in the wild, based on nonlinear mixed effects modeling, and then searching for associated QTL within the genome. Okay. So looking at the quantitative genetics. Looking for associated quantitative trait loci. So what this is essentially doing is using the pedigree in order to interrogate the genome to find linkage regions. So these aren't genes, but linkage regions that are associated. So within any given linkage region, there could be several genes of interest. Okay.
So you had a vervet Dutch hunger winter, basically.
Essentially, yeah. I mean, this was nowhere near as severe as the Dutch hunger winter, right? But essentially, that's what we're looking at. So within these adult measures, this is average body weight, BMI, and whether or not they were chronically obese for their adulthood. Okay? We're seeing narrow sense heritability, the amount of heritability associated with genes, that's on a par with what we see in humans. Okay, so body weight is about 70% genetic, BMI about 65%, chronic obesity about 50%, which is actually kind of high. But we actually can also control for covariates. So we can use maternal ID as a covariate. We can use the diet that their mom is eating as a covariate. And what we end up finding here is that sex is a significant covariate for all of these, which we'd expect. They're sexually dimorphic, right? These are related to the West African species, so very dimorphic. But we're also finding a maternal diet effect, so what the mother was eating when they were in utero. And also a sex by maternal diet effect, which I really wasn't expecting. Okay? We also have some effects from postnatal dietary shifts, but these are not as significant as what we find in the maternal period.
So who is more effective, males or females?
I will show you in just a second. Yeah. So here you can see these are the residuals for body weight and BMI. In dark color, we have individuals that were exposed only to a chow diet during this period. And then in gray, we've got individuals exposed to this high fiber, high protein diet in this period. And you can see that there are there's a little bit of variability postnatally, but not statistically significant. The only place where we see that is in males that were gestated when their mother shifted that diet. Okay, so what we're finding here is that males that were gestated on this high fiber, high protein diet were significantly more likely to be obese in adulthood than males that were gestated on the chow diet. We don't find this in females. Okay? And this is contrary to what we might expect, because if we look at work in rats that look at maternal programming that leads to obesity, it happens in females but not in males.
So that's body weight, not birth weight.
Yeah, exactly. This is body weight.
EW3 to that, yeah.
Oh, birth weight. Yeah, sorry. This is body weight, not birth weight. Yeah, unfortunately, we don't have information on birth weight in the colony. We have about 100 animals that we have birth weight for, but it's not statistically powerful enough to plug into this model. I tried and failed. So this is interesting. Incidentally, I pulled this slide out because there's 6 males in the sample. But if you only look at that first postnatal year when they are nursing, this same effect is present. But again, there's 6 males, okay? So it's not necessarily real.
Yes. So I'm thinking a lot when there's this kind of sex differences about like selection effects, so like the male vulnerability kind of thing,
Yeah.
like maybe when they were sort of the ones that were most able to pack on the weight are the ones that actually survive gestation on the more challenging diet.
Yeah, I tried engaging with that literature in discussion of this paper, and I was like thinking along the lines of like Trivers Willard and the females with allopathic sex, so like males, Tim Kloppenbrock found like cross mammals, males are like more vulnerable to these maternal effects than females are. But yeah, like I just don't have enough evidence to really like plug into that literature. Like it's more hand wavy. Like I think it does fit into that literature maybe, but I think it needs to be investigated longer.
Would like birth rates did they decline during that time? Because that might imply that some of the pregnancies were aborted, and those might have been.
We actually do have data on, and somebody published a paper not that recently on miscarriage in the monkeys. That's a good idea, actually. I'll take a look at that. Because they didn't really correspond to diet at all. They were looking at other covariates. So thanks. That's a good idea.
How dimorphic are they at birth, generally?
Generally speaking, the dimorphism doesn't pop up until puberty.
Not even in terms of body composition? Or
That's something I just don't know.
It's like in humans, there's a little dimorphism, and like females that are gestating a male and eat a little more. And there's some evidence from like supplementation trials and stuff. There's a bigger effect if the baby's male.
Yeah.
So there's some human precedent for this kind of thing.
Yeah.
But yeah, I don't know about the long term. What did they find with the Dutch famine? The original, obesity finding. Like, there's been a bunch of stuff since Doha, but there was this finding, forgetting what year it was. This published in like New Journal Medicine or something, but they showed this obesity effect. And I feel like there might have been a sex difference there.
Okay. I'll check that out.
Yeah,
Like, I know
it's not real fresh from me right now. I can't remember. It was a pretty robust finding.
And was it male or female?
I think it was in primarily males.
Okay.
But in some of those.
So that would be great.
Yeah.
So I'll look for that. Because yeah, I really
It
struggled
was first trimester
this.
specifically. It was not the other. It's like early
Yeah.
gestation.
Okay.
Or obesity. I think it was specific.
So I also tried looking at the trimestral effect, and
It's
again, the sample size is not large enough.
too much.
Yeah, like we do know what trimester they were first exposed, but I would then struggle to understand how much of it, does the trimestral effect matter when they're experiencing this altered diet throughout their developmental profile? And that's something that we can't address either. As the vervets continue to grow, we are planning on doing some dietary intervention studies, in which case we hopefully will get DEXA at various stages. There's also a little bit of talk about maybe sample like amniocentesis in order to get at what the actual internal environment is for these babies. But yeah, it's still in the planning stage. So hopefully we'll be able to kind of suss out some of this, but for the moment we can't really address that. So interesting findings from adult onset obesity. But then I also dug into those growth models. So I used nonlinear mixed effects to characterize growth. And on top of that, I used a clustering algorithm, like a KLM clustering algorithm, in order to kind of suss out what an obesogenic growth profile actually looked like versus a non-obesogenic. And rather than saying, this is what an obese adult is, and this is what a non-obese one is, sort them, I essentially let the clustering algorithm do it agnostically. Because of course we do have animals that may have this obesogenic profile that didn't actually meet our standards for obesity. And there may be other animals that have a non-obesogenic growth profile that maybe had a heavier growth weight that then wound up in that range in adulthood. So we used the growth pattern to suss out obesogenic versus non-obesogenic rather than the outcome. Then I created growth models, again using nonlinear mixed effects modeling and assessed heritability of the parameters of growth. So just as an example, here we've got the growth models for body weight. In dark blue, we've got males that fit into that obesogenic growth cluster. In light blue, males that fit into the non-obesogenic. In dark yellow, we have the females in that obesogenic cluster for females, and the lighter females, non-obesogenic growth pattern in lighter yellow. And here we've got the three parameters. We just use a simple logistic growth model, which I know is not ideal, but when we've got samples that are only occurring 3 times a year, this is about as good as we can do. Because the only time we actually are measuring these monkeys is when we have what's called the roundup, which basically, because of the federal regulations, we need to test them for TB 3 times a year. So during that time, we just collect this phenotypic data, because they need to be anesthetized anyway. With asymptotes of growth, so this is where growth levels off, we see what we expect. We've got clear sexual dimorphism, right? Females lighter than males. And then we've got these obesogenic trajectories leading to heavier body weight, right? It's not revolutionary, right? It's where we get into the other parameters that it gets a little more interesting. So here we've got midpoint of growth. This is essentially a measure of the duration of growth throughout the lifetime. And within this case, we've got clear sexual dimorphism and duration, and this is what Steve Lee found also with his growth models. Sexual dimorphism in a particular species is largely the outcome of binaturism. Males are just growing longer than females, and that's why they get bigger. But even within this, there's a slight but statistically significant difference between obesogenic growth and non-obesogenic. So this obesogenic growth pattern is one also of longer growth. So they're growing longer, and this is again making them much larger. Within rate of growth, we're seeing that there is a statistically significant difference between obesogenic and non-obesogenic that's not sex-specific. So we're just seeing that these obesity trajectories grow slower over a longer period of time. So they're packing on the weight not because they're growing really quickly, it's just attenuated throughout the growth cycle. I don't know what to think of that either, because that doesn't jive with human literature.
So let me see that. So obesogenic animals are growing longer.
For longer,
And
yes.
they actually going through like NRP and stuff later.
This is something that we can address.
It's the opposite in humans, right?
Yeah.
And you expect the kind of acceleration of all that.
Yeah, exactly.
But it's growing longer in mass. What about skeletal maturation?
We also did this for crown to rumpling,
Okay.
and the crown to rumpling models don't show the same pattern. And so this does seem to be them packing on weight rather than growing weight. Yeah, exactly. There's like a little bit of a signal where these more obese animals are getting slightly larger, but that's not necessarily unexpected because when you dig into the literature, there's a lot of pleiotropy between skeletal and body weight growth as well. So this does appear to be largely tissue growth, right?
Was there,
Yeah.
I don't know if you can call it a model, but was there interactions between certain development periods, like?
Yeah, we really couldn't say. We did look for like dietary effects at different periods, but this is just divided into those 4 periods of dietary exposure. Because we only get it 3 times a year, these like more finely scaled developmental periods, we don't have the resolution to look. Because they're probably happening like in between. Because I did try to actually look for inflection points with more finely scaled analyses, and we don't have the resolution. So it seems like if there are any spurts that are happening, we don't really see them. We'll see like, if we catch the right window, we'll see a larger difference than, a window that's a little closer. But again, this is kind of cross-sectional, so it's not ideal for that.
So the growth model that came out of this was just talking about duration of growth and rate.
Over the lifespan, yeah, exactly. So again, like not a very finely tuned model. Like ideally we'd use like a Jolly model, right? Or even something like Gompertz maybe, which we've seen in other animals. They can give us a little more resolution about these inflection points that we would expect at different ages. But yeah, the data just isn't finely tuned enough for that, at least over the lifespan. We really have to sample kind of like twice as frequently in order to get something like that, unfortunately. There may be more resolutions. There's a paper that will come out soon that I'm collaborating on with Jonathan Jarrett, who's a student of Peter Henze's. They have a field site in South Africa where he's managed to train monkeys to walk onto a scale. And so he actually is getting more finely tuned growth and can use these like more refined models. So I think that is going to be really interesting when he can get that up and going because right in his dissertation. So maybe not anytime soon, but in the next couple of years, I think we'll be able to address that a little better. So assessing the heritability of these growth parameters, again, this is the asymptote, duration, and rate of growth. Asymptote, fine, it's like very similar to what we find for adult body weight, but with rate and duration and rate, we're finding some genetic heritability, so there's some genetic signal for these variabilities in growth rate. And then when we actually look at the covariates, we're finding the same that we found with adult phenotypes. So these growth durations and rates are also being influenced by this sex by maternal diet effect. So not only is this dietary shift having an impact on overall adult phenotypes, it potentially is having an effect on those adult phenotypes by affecting growth, which is not revolutionary, right? We already know this happens in humans, but it's good to see that it's happening within this model system. When we actually dig into this more, so the quantitative genetics is great, let's find genomic regions. We can use that pedigree using something called a multi-point linkage analysis. This essentially is using the pedigree to strengthen the statistical power of actually finding linkage regions in the genome that might be associated with these traits. And what we ended up finding when we looked across the entire genome was we had no genome-wide statistical significance, which was a bummer. But we do have a lot of genome-wide statistically suggested factors. What this says to me, because I'm an optimist, is that when we increase that sample size, which we do, there's 50 new babies born every year, and you can track the growth, we may be able to kind of push this up to genome-wide significance. But suggestive in many cases is kind of enough to point us in a very interesting direction. So we did get a number of hits on various chromosomes. And when you actually dig into those linkage regions, because the vervet genome, you can find it on an ensemble that's fully annotated, so we've got all the orthologs with human genes. What we ended up finding are hits in linkage regions that do have a lot of loci implicated in type 2 diabetes, in insulin dysregulation, in obesity writ large, in thyroid disorders, various metabolic disorders. And what this suggests to me is that this actually is reflective that this obesogenic system that we're finding in vervets is largely in line with the obesogenic system we find in humans. Because we're getting the same hits correlating with the same phenotypes in this admittedly rough, but still interesting analysis. Yeah.
Just really quickly, was there any difference, just going back to the diet?
Yeah.
Any difference in gestational age or things like that?
In what? Oh, gestational age of birth.
Or twin, I don't know, anything about.
Yeah, unfortunately, we won't get gestational age of birth because all they do is to get at transtral age, all they do is extrapolate back by the standard gestational period because they don't have a good sense of when conception actually occurs. Because births happen more or less year-round. In the wild, they can be seasonal, but in the ERC, it's a little more scattered, as often happens in captive populations. So yeah, we don't have a good sense of what their gestational age of birth would have been. I also was really interested, again, like looking at birth weight to find out if like catch-up growth is the thing that's happening here, and I think that's might be what you're getting at too, indirectly maybe. But yeah, it's not something we can get at, unfortunately. It's something we could if we like, pay for it, right? We could pay them to actually find out when they're getting pregnant and actually get a gestational age, but this is not something we've done so far.
Yeah, okay, so you've got the human phenotypes these are associated with. Do you know that the functions that they're...
No. So, I mean, they are annotated for function. And there's a supplemental table in the paper that also lists the annotated function. There's a lot of kind of like lipid transport and various other, like that was the one where I was like, oh, this is interesting. But there's many other genes with other functions that I wouldn't know how to actually correspond to that. So I'm not a cellular biologist.
Because if you look at the log study we were talking about, which is a big genome-wide meta-analysis of GWAS studies in humans predicting BMI. The overwhelming story that comes out of that is that those genes are coding for hormonal traits, right? And they actually do a transcriptome. They look at all the publicly available transcriptome data, and those genes are overwhelmingly being expressed in the brain. And there's a bunch of studies now that show trade-offs between the size of brain structures and BMI. And so there's something really interesting going on there. We've argued that it's probably an energetic trade-off possibly. I just wonder, it's one of the things that might be sort of unusual about humans and maybe make it hard to extrapolate to other species. Is this, large brain size and so forth. But that's the main story that comes out of a lot of paper, is that it's about the brain.
See, that's interesting because like the papers that I've read, at least, so when I was like doing reviews trying to find out what regions would be interesting, like a lot of what I read, at least in monogenic forms of obesity, is that like some of these neuronally expressed genes, like MC4R would be a neuronally expressed trait, and it usually associates with a lack of appeasement during eating or hyperphagia during eating.
Right. That's the assumption that people have, is that it's about behavior. But if you look, there was, I don't want to get too off track here, but there's that meta-analysis, 97 hits that they found. They generated a polygenic risk factor score, and then they used that to predict BMI, but also cortical gray matter volume, and the relationships are equally strong in opposite directions. And in fact, gray matter volume mediates the relationship between the polygenic risk score and BMI. And so gray matter volume is what makes the brain costly. Right? So there's probably some kind of behavioral component. There's got to be a energetic component as well. Anyway, it's something to think about. Like this might be one way that vervets are different somewhat.
And I'd really like to find out how they might work. That's going to dictate how useful this is as a model, right? I mean, I've been mostly digging into it, like at least in the wild sample, we find a lot of pleiotropy also with like circadian rhythms, so like the timing of eating, and also how the timing of eating interacts with like response to temperature. So there's a whole genomic system that kind of links changes in temperature with changes in circadian rhythm, especially as regards when animals are eating and how that actually influences how they deposit caloric calories into the eat. So like, yeah, I've been struggling too with like how to actually kind of bring all of this into focus as a systems biology question, because there are so many systems that are implicated. It's really confusing really quickly.
Because the links between obesity and metabolic phenotypes is one thing.
Yeah.
And that could be the same across rats and mice or whatever. But what is it that actually drives, what is the dominant factor that sort of trade-offs that drive excess weight gain? It might be quite different. It might be related to differences in, appetite regulation around circadian rhythms, around species and brain and another one.
Yeah, exactly. Yeah, so that's something that I'm not quite there yet. No one is. No one is, yeah. Okay, so just to summarize, like I got to look there, but I only like 10 questions, right? In captivity, we find this is partially a developmental process of epigenetic control. Individuals exposed to dietary stress in utero, maybe when nursing, become significantly heavier both relatively and absolutely as adults. And it looks like there's multiple loci involved that are associated with obesity-related traits in humans, and these are detectable. So I'll spend the last couple minutes that I have just talking about what we're doing now and how we're trying to push this forward. In the VRC in particular, we've got a plan that we need to get funding for, so this is all also generated. But I think it's an exciting way to go forward. We're going to continue with the growth modeling and measurements and further dietary interventions within captivity. We're going to eventually have enough power to look at age and sex-specific variability in QTLs, to find out if females and males are becoming obese in different ways. Exposure to different ages are part of that. With Kylie Kavanaugh, we're going to look at DEX phenotypes, so we are going to be able to look at how fat is being deposited in the body at different ages, whether or not those deposition patterns are different according to dietary shifts. RNA-seq for gene expression in adipose with equivalent, and then Katie, they told me they get involved in the VOC. I don't know if that's true, so I threw that up there, but I'll let
Yeah, it is.
It
We'll
is.
say it is, yeah.
We'll say it is.
Sometimes it's just samples too, yeah.
Okay. So maybe Katie is involved in the
I am biting.
What's that?
I am biting.
Okay, good. So I'll keep that in there for now. And then taking this back to the wild is what I'm especially excited for. In the field, I have focused on two field sites in South Africa that we've sampled at broadly, both of them relatively close to Bloemfontein and Free State. One is during nature reserve. This is a group where the males may migrate in from farms, but within the nature reserve itself, they're very rarely provisioned. And then farm on the Green Dam, the Southford Stud Farm, is a horse breeding farm, and monkeys on this farm have access to peanuts. They also have access to horse feed, which they eat very liberally, and the horses can't do anything about it with their horses. So they get a lot of provisioning. And the really good thing about the samples is that there's been drought in South Africa. So drought, we already know, is a really strong selective force. It leads to lower availability of water, lower food availability, and then on top of that, the plants that experience severe drought, work done by Jessica Rothman and others, has actually shown that they produce less calorically valuable foods. So the foods that you get from a drought-affected plant are less nutritionally viable than fruits from non-drought-affected plants. And this actually lasts for quite a long time even after the drought is abated. And sorry for the sad picture, but we do know that primates are really strongly affected by droughts. This is a really strong selective force for them. And of course, I have to throw in the slide about how drought is thought to be a selective force in humans. This was the idea behind using PIMA as a model for evolution of obesity. They do have very high rates of type 2 diabetes, and this was proposed as a potential example of thrifty, either genotype or phenotype, depending on which literature you're reading. And so this is kind of a way to gesture towards the human literature, a more explicit evolutionary model of potential obesity. And within South Africa in particular, a drought occurs every 10 to 20 years, and there's typically several years in duration. So there is actually very deep history of occasional drought here, just like what people hypothesized for Pima, for example. And the most recent drought began in late 2010, early 2011, right after my postdoctoral sampling of these populations in South Africa. So we have many samples from these populations just prior to the beginning of the drought. The drought peaked in 2016, which was my first collection season as a professor at BU. So we got a peak drought sample. This was the driest year on record in South Africa. And this is just a picture of during nature reserve. This is during that peak drought season, and this is the most recent yield season. And so you can see the river, at least, is much bigger. Whether or not this means the territory is better, that's something that we can't really address right now, but I will say that the monkeys looked a lot less dying in that yield season than they were before. So I'm also interested, though, in how human impacts interact with ecological stressors like drought. This was a really productive conversation with Tom yesterday. We went and grabbed a beer and talked a little bit about this. Because the vervets are experiencing very variable stressors depending on whether they're living with humans or not. Okay? So in during nature reserve, again, they're really affected by the drought. They're running out of food. When we trapped them, we also collected air samples to kind of buzz their air that they put back. I went back this past winter, South African summer, and they hadn't thrown back their air yet. Like that's how nutritionally stressed they were. They just were like not recovering. Meanwhile, in Korea, we went there, like everything had grown back, all the monkeys were doing great. They're like eating horse food. It's wonderful. So that's great, but in Sitturing, they're really buffered from humans. Like humans will drive through like touristy things, but they're not being hunted. But here in Harib, there's a lot of anthropogenic impacts because the farmers aren't very happy about the monkeys stealing food. Now, speaking of this kind of nutritional buffering that's happening on the farm, we do find that skin-fold thicknesses are significantly thicker on Harib than what we find in Sitturing. And this is during this peak drought season in 2016, 2017. So this is really great evidence that there is some nutritional buffering happening on the farm as opposed to within the nature reserve. However, these kind of traditional clinical indicators of body condition, we're not really finding it signal here. The females seem to be fitting in with what we hypothesize better than the males, and again, I think this might be an artifact of males coming in from farm areas. And so not really having the phenotype you associate with starvation conditions in that case. Again, there is a trade-off. Like I like showing this video because it really kind of exemplifies what happens when humans don't like monkeys. This is Lieutenant Dan. He has had both of his legs amputated by snares, and there are several other monkeys here who have limbs amputated from snares, electrocution scars from power lines. We find often monkeys that have grape shot or buckshot stuck under their skins that they've been shot at and then subsequently healed. So there's a lot of stress aside from nutritional stress within this colony that we might have to contend with. So my postdoc, Alicia Rich, is kind of trying to dig into the gene expression literature to understand how we can kind of suss out differences between nutritional and this kind of psychosocial or ecological stress. And so we kind of have a nice, kind of natural experiment where we can look at the effects of drought with this pre-drought and peak-drought sample, and we proposed a post-drought sample on the drought's dating. And we also have this gradient in human impacts, so the kind of stress that they're receiving because of being close to humans. And what we're hoping is that we can do differential gene expression analysis, and in that way find networks that are up or down regulated across these gradients. My hope in relation to the evolution of obesity is that in these drought segregated conditions, and also the dietary buffering that kind of goes along with them, that we'll be able to find networks that would be implicated in the evolution of obesogenic traits. Because again, we're not going to see these guys become obese in the field and never going to get enough food. But those traits are presumably there, and hopefully they'd be detectable within the system. Hopefully. We'll see what the funders say. And we also were considering looking at trait-associated genes. We found a really great study of sea lions in captivity, where they actually used trait-associated genes associated presumably with either nutritional stress or psychosocial stress, and they found really good differentiation when they experimentally induced both in these sea lions and used this trait-associated gene protocol to find out and to actually correlate it with these two different kinds of stressors. So we're hoping that this is another kind of method that we can use to suss out the differences between psychosocial stress in the captive condition versus this kind of ecological buffering from drought. So, related to these goals, we need to sample some more, particularly males on the farm. The males on the farm are like really savvy about the traps, and they're like, so our sample for the males on the farm is a little low. We're going to keep trying to do that, and we have to go dart them because, they're already really wary, but they're not going into the traps. So we need to increase that sample. I do want to track this potential male dispersal effect, find out where these males are coming from to see if they are being dietarily buffered during ontogeny, if this is interrupting that signal. And also, like I mentioned before, we really do need to look more closely at what they're actually eating and what kind of calories they're getting. So some nutritional ecology data would go a long way towards making this a little more viable as a model. And in the meantime, I'm developing this Smeagol lab. Alicia, my postdoc, is going to be working on this differential gene expression experiment. We're collaborating with Amita Kapoor at Madison. She's in Tony Ziegler's lab to pull out not only glucocorticoids to look at stress, but also leptin and also sex-based hormones. She's got an assay where she can get 8 hormones at one side of the hair follicles and then look at it longitudinally, and it's super cheap. So FYI. Good person to get in touch with. And then I've got a grad student, Christian, and he's digging into this populational data to suss out, at the beginning, just some variation in the UCP1 gene region. UCP1 is the protein that causes non-sugaring thermogenesis in brown fat tissue. In humans, we do see latitudinal selection for UCP1, so we're just starting off nice and easy with the genomics, looking for a signal of selection within our vervet sample. Yeah, so that's what I've got for you. Thank you for the invite, Stacy, and I'm happy to answer any questions or talk